Yeah, we're gonna, this week probably, we will have a time, one of these periods we'll use to get everybody together. Get everybody together means, you know what I mean? And then we'll explain we'll share the videos first and then we'll do the reflection. Or explain how to do the reflections. Okay. Right, so good. Oh, my stuff is here. All right, so let's try to finish digestion. Oh, that's annoying to have that there, isn't it? That's okay. I don't want it to record right now. No, I don't. Should be a little bit of both.
I think this will be better.
I trust you. This, I want it to go away. What? Oh, I know. No. Okay. Careful. That's not mine, that's Mr. Two's. You'll probably kill me. It's broken. All right. So we'll try to finish digestion here. Thank you, Richard, for your help. And like I said, so that top of the diagram there, we should have had for last lesson. So you can use that to help kind of test to see your how much you remember the labels and the annotations of the villus and how it's adapted to its function. Today we'll focus on the bottom part there. There's a little bit of cutoff, but I think you know what it says. So this involves a bit of review from topic 1. Okay, so membrane and membrane transport. So now suddenly it's freezing cold. We go to extremes. I'll just turn it off for a second. So that temperature.
Which PPT is that?
Digestion 6.1.
Are we reviewing?
Wait, did we already do? We didn't do the last part of this, right? We didn't. Oh, didn't. I did it with the other class. We haven't finished. We haven't done this, and this, and this, right?
No.
Okay, I just want to. It's possible that I've made a mistake, but I don't think I have. Okay, so what we're going to do with the last little part, this part is fairly new, okay? We're going to look at the very smallest unit of the villus, which is one epithelial cell. So if I go back, back to here. So here we can see this is each one of these is an epithelial cell, right? Like one. The dark part is one nucleus. So that's our single cell sort of wall. Single cell thick wall of the villus. Yes, thank you. You also have, this is just an extra, you have some goblet cells. This provides some mucus or maybe some enzymes. But in any case, we're going to look at like a couple of these cells really close up. First thing we'll notice is if we look at a micrograph, we see that the edge you can see the microvilli much more clearly, okay? Compared to here, you can't see the microvilli along the edge, but here you can. It's almost like a toothbrush, right? So you can see it's like, or like a spiky haircut, sort of. So the microvilli here form what's called the brush border, okay? Very appropriately named. And as I mentioned previously, the brush border enzymes, yeah,
Jay's trying to get in.
thank you. Now here's the question, where? Oh, there. Why is this here? Can I not move this to here? Yes, I can. Amazing. And you can still see. What just happened? See, like, why would that happen? The zoom is here and I'm staring at it. When I click participants, it goes there.
It's because you want to pull the participants so that if you pull it back, it will get it.
Why? It's so stupid. Just keep it together. All right, so what I was saying is these epithelial cells also will produce some enzymes. Remember we said, like, yes, some is produced in the saliva, yes, some is produced in the stomach, and then yes, a lot are produced in the from the pancreas. But in case that's still not enough, you will have some that are kind of just sort of like stuck in the microvilli. So it's produced and just kind of sits along the edge, so that any food that comes past that hasn't been fully digested yet will get digested fully along here, which can then be absorbed right away. So it's like a last resort. So you'll see some extras like maltase for maltose, dextrinase for those limit dextrins. Remember a ways back we said, like, these sections that don't get fully broken down, those will need an enzyme like dextrinase, okay? And then some other lipases. Endopeptidase is just a fancy word for what? What do you see in it? What is this breakdown, endopeptidase? Polypeptides, yeah, proteins, basically. Right, so you could say protease instead, if you want. And nucleases for like DNA and things like that. Okay, so all of this stuff that might not, sorry?
It's really nice stuff.
What's stuck? Doesn't seem stuck to me. Oh, I think I got kicked out. Oh, yeah. Somebody tell them I just got kicked out. They don't have their phone. Just send them that I got kicked out. It's reconnecting. Okay. The only thing that's left that doesn't digest is cellulose, right? So cellulose, another word for cellulose, more common term is part. Say again. Wait, according to this, we're not connected. Who's speaking?
It's Amanda.
That was Amanda? Really? Didn't sound like that. Sounded like it was coming from the speaker. Okay, sorry. Sorry, Amanda, what did you say? Oh, you think it's wrong. Okay, what's another word for the stuff that's not digested? What is happening? The food that you eat there we go, that doesn't get digested. What part of your food can you not digest?
Fiber.
Corn.
Is that okay? Corn, what is corn? Okay, corn, what's corn mostly made of?
Oh, fiber.
Thank you, fiber. So cellulose is really just fiber. Right? It's the stuff that makes up the cell wall. Okay, everyone okay with that? So just to like an additional point is that there still are enzymes along the wall of the small intestine. Okay. So some review that you are going to want to do, or that we should probably do.
Oh no.
I can't hear anything.
Digestion, so perfect reason to dip, you know.
Why is Jay at home? Sounds like a good choice for me.
Now, can you hear us now?
You should editing.
Wait, no, where's the new one? What the heck? Oh, you're still there.
She just muted.
Tell them we can hear them.
No, because
Michael said he just muted himself.
Well, hello?
Yeah,
Hi.
we can hear you guys.
Oh,
We
okay.
Cool.
Well,
just simple.
okay. Cool.
We can't see anything, but okay.
Yeah.
You can't see anything.
Yeah.
Can you just put me in so I can know what's happening to them?
Like, in front.
Oh, I can see something.
I'm gonna spend exactly 1 minute trying to do this, and then I'm done. Now,
Yes.
Yeah, but it's super lag.
well, that's not my problem at this point. Sorry. Okay. So we need to review or have some knowledge of the cell membrane. Okay. This I can't ask this question because I just gave you a handout with the answers on it. The different methods that the now digested nutrients can get through the plasma membrane will be different for each one. So they don't all use the same way of getting in. All right? We need to be familiar with these modes of transport. Okay, so again, if you don't know what these are at this point, that's from a different topic. So we're now going to refer to these. Okay, if you're not sure, it's something worth revising. So of course, osmosis diffusion, facilitated diffusion, active transport using energy, endocytosis using the membranes. Okay, we're going to talk about all of these things today. So, first, let's look at, let me say this. The example that you need to know, need to be familiar with is, according to the learning objective, is describe how the modes of transport, or how the breakdown products of starch are transported into the bloodstream through the epithelial cells. So meaning, glucose. Okay, so the only example you need to know is glucose. I'm going to show you what happens with fatty acids. I'll show you what happens sort of with proteins, amino acids, but the only one we are going to go through in detail is glucose. Okay, so here you can see how are foods broken down. So again, we're specifically just focused on the three main micronutrients. So first, what's interesting is fatty acids. Okay, this is the yellow ones here. They're able to diffuse directly through the membrane. Just bam, right through. Why do you think that is? Two different reasons, actually. Why do you think that is?
No, polar.
Try again.
No, polar.
They're non-polar. Wait, no.
The membrane polar or not polar?
It's
Partial.
polar, right? The fats are polar, which helps because the membrane is polar, so it can kind of orient its way through preferentially and get through the membrane. Okay? That's one thing. Or if the fats are larger in size, you can use a vesicle. What's this? What are these pink things? What is Yay, Golgi apparatus. You're close, sort of. Not really. Golgi apparatus, right? Yeah. So remember, Golgi apparatus is where you get vesicles being formed to secrete things by exocytosis. obviously it can also be used to bring things in. So larger fat globules or larger triglycerides might be able to use endocytosis. But most, if they're small enough, will diffuse just through the membrane itself of the epithelial cell. Now the problem is for glucose. I should mention amino acids first. Amino acids have a very diverse mode of entering cells. Why is that? Why are they why can't we describe just and say like this is how amino acids enter the cell and then the bloodstream? What is particular about amino acids? R groups. What about the R groups?
Different shape.
There are a lot of R groups. Maybe not a lot, but specifically how many R groups?
22 or 20.
We accept we say 20. Yeah, so 20 different R groups means they're all different, right? Different charges, different polarities, different hydrophobic, hydrophilicness. All these things are different. So you would have to spend like different lessons explaining each different one. Okay, so we can just accept it like amino acids some will diffuse, some won't, some will need protein, some won't, some will, yeah. So there's a variety of ways that amino acids get in. The one we will focus on is glucose. So here we can see at the bottom, glucose is polar. Because it is polar, because it is polar, it can't diffuse through the membrane. So then that means fatty acid is non-polar.
Yeah.
Yes. Oh, fatty acid is non-polar because it's just a long chain.
Yeah.
Yeah, so it's all even.
Wait, so the fatty acid and amino acid are not included in the
Let's say that, yes. The only one that they would ask you a question about is glucose. Okay? So we say that glucose is polar. Because of its polarity, it can't diffuse through the membrane, so it needs a special way of getting through. Now, while we're waiting to explain that, you're going to want to draw, and you can just use either the back or you can use your jar book or something. What we're going to draw is this. It's basically the villus. Okay, let me finish the first part. Okay, so it's like the villus, okay, that we did before, but I'm just going to exaggerate like two cells, okay? Two really big cells. Might make it even bigger so it's easier to see. Okay, so what we're showing here is like, does everyone see what I'm trying to do here? Yeah? So it's like if we took like two of these, like this like little cell, right? And then we just made it big.
Looks terrible, but
It looks awesome.
manually zooming.
Yeah, exactly. Okay, so we're looking at the edge of, I guess you could show. Anyway, I'm not going to get too carried away. Okay, so go ahead and draw. I'll give you a couple seconds to sketch that out, and then we'll start into how does glucose actually move into the cell. I'm going to draw not too much over here, mostly inside this.
wait, but I remember in IG it said glucose are being absorbed through diffusion.
Yeah, fake news. Guess
Yeah.
it's tragic. Is it working now, people?
Yeah.
Yes.
Okay, sweet. Sorry. Is this thing I need a charger. Why? Okay. Right. So hopefully we've got a bit of something sketched out. What we'll see here is for HL students, we've seen something similar to this before. Okay, twice actually. We've seen it once in root hair cell, and we've seen it once in kidney. Okay, very similar concepts. Essentially, like what Richard was asking is, didn't we say in IGCSE that it's just absorbed, like it just moves by diffusion? Yes, I may have said that. I also may have been simplifying it so that you just learned something enough so that you could not have your brain explode. But the truth is there's obviously a lot more going on behind the scenes. So there's a sequence of events in order for glucose to be absorbed. Okay? Now like any kind of sequence that's fairly cyclical, sorry, we should also add a few blood vessels here, I suppose. Some blood capillaries. Okay. Because ultimately, what we're trying to do is to get the nutrients into the blood, right? This is the goal, to get blood into this place. So first, let's label our areas. How does this look? Does it look okay? Nice. All right, out here, what do we call this space outside the villi? Lumen. Yes, lumen. Good. So what we have, sorry Amanda, I lied. I am going to draw a few things out here. I just want to say that, so in the lumen we have the food, right? The food that has been digested. So you have things like glucose, you have amino acids, you have fatty acids, you have glycerol. I'm not just going to ignore glycerol for now. So I'm just going to draw many of these just to show that like they're all here, right? They're all floating by. So they're outside. We want them to be in here. So I'm going to pre-draw this. So this is my end goal, right? The goal is for glucose to be in the bloodstream. Once it's in the bloodstream, we can pump it around. Sorry, first it goes to the hepatic portal vein, then to the liver. Then it can be pumped around the whole body, right? So that all the rest of your tissues can use glucose. So this is the end goal. It starts out here, ends up in here. Bonus question. Make sure I have time for bonus questions. Ready? Oh, the clock is gone. That's why I own this watch. Bonus question. how many, we asked this before when we did the like respiratory system, how many membranes would glucose cross to get from the lumen into the bloodstream?
Three.
Think about it for a second. I'm going to ask you in 10 seconds to hold up the number of fingers. How many membranes does it pass through? 3, 2, 1. How many membranes? You're so scared. Like, some of you are like Snow White, like witch. I see a range from 3 to 5. I guess if you took the average,
Four.
you'd be right. Yeah, 4. Why? Because what's the capillary wall look like? What does the capillary wall look like?
One cell.
One cell thick, right? So actually this is like
Or
this.
two membranes.
Is like this, right? The wall of this is one cell, which means 1, 2, 3, 4, then inside. That's just bonus for challenge. Okay, so now, sorry, I was supposed to tell you to draw this in pen because you're gonna have to add some things. I mean pencil, because we're gonna add some things. All right, let's add a few things. The reason we have to recap membrane transport is because all of this is going to involve membrane proteins. Remember fluid mosaic model? The mosaic is that there's proteins stuck in the membrane. They float around and move around, but they help to move things in and out as well. They act sort of like gates or doors or channels or these kinds of things. So first, let's do, I want to try to stay consistent. Okay, so let's do over here on this end. We're going to add a sort of like a gate. Okay. We'll do that one in blue. Okay, so you'd have many of these over here. We're only going to kind of refer to one, but we can say that there's quite a few. Let's add them all now, and then we can just talk about them later. We're going to add another protein on this end, which is going to be more of a channel. Okay, so it's going to be an open, like this. Let's add one more here.
Now, what is this drawing?
The name is epithelial cells of the villus. Or you could say absorption of glucose in the villus. Absorption of glucose by the epithelial cells of the villus. Is that good, descriptive enough? Descriptive title. Okay, and then one more is going to be on the lumen side. Oops. On the lumen side is going to be another several proteins. I'm going to do it sort of different shaped here just to have some variety.
Microvilli or villi?
I said villi because this is all part of the villus, right? Microvilli really only refers to this folding section. So it's all, like the whole villi is covered in this, so the villi is more appropriate to say. Okay. So I don't want to like ruin my drawing too much, but the first place we're going to start is with this blue protein. Okay, this first blue protein is one that we've seen before. How do I keep this like? Okay, I'll do this. No, not yeah. That's This blue protein is called the sodium. You want to finish my sentence? Sodium, potassium, what? Not channel, but pump. Oh, not bump, pump. Sodium, potassium pump. Yes. The sodium potassium pump. What is the function of the sodium potassium pump? Or what are some features of the sodium potassium pump? And potassium. Wow, yes. That is accurate. Can we be a little bit more specific?
3, 2 both directions.
3 and 2, yeah. So there's a difference in which way the ions are going. So if you look there, you can see that in our case, 3 sodiums are being pumped in one direction, and 2 potassiums are being pumped in the opposite direction. In this case, you get 3 Na plus is your ion sodiums pumped out, and 2 sodiums pumped into the epithelial cells.
But how would that balance out?
Doesn't have to balance out. That's just the way it functions.
But if 2 goes in and 3 comes out, eventually wouldn't it run out?
Yes, but you'll see why not soon. It will fix itself. The second question, when we say pump, this is pumped out, it's pumped out. What does that imply?
Energy.
Energy. ATP required. So in this case, we say that, or we must assume that regardless of the gradients, those 3 sodiums go that way, potassiums go the other way, we spend energy to do this. We don't care about high and low concentration. Okay? So I'm just going to do... This is why I didn't really want to do an arrow, but okay. So we can say that, maybe I'll use black for this. So 3 potassiums are pumped out. Okay, 3 potassiums are pumped out, while 3 sodiums are pumped out, while 2 potassiums are pumped in. And this goes on at all of the cells of the epithelial, right? Like, so if you wanted to continue, you could show more of these everywhere along the edge, right, all the way down. This would be happening. Okay? Like this. Right? 3 Na plus 2 K plus. Oh, wrong way. Okay, if that's going on, what does this create in this cell? What is the environment in the cytoplasm of that cell now? In respect to these 2 ions?
Lower concentration.
Of?
Wait. Higher concentration of water. Wait.
And? Wait, what? If this happens over and over and over, what is the the ion situation in this cell?
False. The
It's
overall ion concentration should fall, right?
okay. Don't ignore overall. Specifically for these 2.
So potassium, right?
K plus concentration is increased,
So they're decreasing.
decreases. Okay, we're gonna ignore this one for now. Okay, this one is important. We've created, remember HLs, the fake gradient, right? We have created a gradient now. So now, the concentration of sodium ions decreases inside the cell. Okay. We're gonna kind of like ignore potassium. We're gonna stay interested in sodium. That's why you don't really see potassium right now. Also notice that this is like flipped version of that, just so you. Sorry, question?
Can you take a picture of that and send it to us in chat? Because it's really fun.
Yes. When it's all done, I will. Oh, actually, I don't need to be like doing this. Let me try one thing. I don't want to waste too much time. No, that'll happen later. It's okay. I'll send a photo later. Okay, so now we're going to use this invented concentration to our advantage. Okay, if the sodium concentration drops here, then what we will do is we will use the fact that this is lower, okay? It is also lower than what's going on out here, because assuming you're eating a bunch of like foods, there's going to be some sodium ions in the lumen as well, right? There's going to be some sodium hanging out here amongst all kinds of other things. So there'll be sodium here. So relatively high sodium outside, low sodium inside because of this pumping action. Yes.
Like tilt it up so we can see half of it. Okay.
Okay. So now that you have a high to low, now there's a gradient of sodium high to low to inside the cell. We're going to use that gradient to power the entry of glucose. In order for that to happen, we need this red thing. And this red thing, if I can add a label, let's add the label here. No, that's not enough. I'm going to go to the other side. This is the sodium glucose co-transport protein. Sodium glucose co-transport protein. What does that sound like it's going to do? Co-transport. Help, assist, also facilitate. It's going to be an example of facilitated diffusion. Yes, if it's sodium glucose co-transport, they're going to transport both together, right? They're going to transport together. So what we'll see is this. The co-transport protein allows for both of these molecules to move at the same time. So they allow a space for glucose and a space for sodium. But it's kind of like only if sodium is moving is glucose allowed to come too. Okay? So if sodium's going to move, it has to have a gradient from high to low because it is an example of facilitated diffusion. Okay? Facilitated diffusion, which just means it's diffusion, so the movement of high to low, but through a protein, okay, instead of just through the membrane. So it's going to use the fact that sodium's going down the gradient to bring glucose with him. Okay? It's like sodium has a ticket and they have an extra ticket to get in. And so they're going to bring their friend glucose. Okay? If glucose tried to go by themselves, they can't go. They're not allowed. They have to have their like VIP buddy. All right? They have access. So this movement of sodium down the gradient powers the glucose to come with them through the exact same protein. So there's like a space for glucose, a space for sodium. They can go in together if they're both there. Okay? So it's facilitated diffusion, but requires, I'll say he uses the sodium gradient to bring glucose and sodium in to the cell together. Okay, so without that gradient that was created here, you don't get this co-transport. So we'll get glucose, which can go in, all right? Let's show it going in here. At the same time as sodium. Okay, last but not least, tell me again, what's the environment now inside the cell in terms of glucose? High concentration. If it's constantly being brought in because of the sodium gradient, you now have high concentration here. But then again, when we say high concentration, what is it high compared to? It has to be from compared to outside or compared to the bloodstream. It's going to be higher compared to the bloodstream because this is always pumping, right? We said last time there's always movement of the bloodstream to keep this gradient maintained. So now you have got a high glucose here to low glucose here. How do we allow the glucose to move? Even before this, we said it's not allowed to pass through the membrane because of its polarity. So it needs a special channel. So you have, on the other end, a glucose channel. Glucose channel protein. Right? This is going to be I need a new green marker. Facilitated diffusion. Right? Glucose moves. Oh my goodness. From high to low concentration into the blood capillary. So we can show that here through the channel. No energy required. That channel doesn't even open and close really, but it only allows glucose through. Nothing else can go through that channel.
But if glucose is polar and cannot go through the cell membrane, how does it go past the capillary cell?
We're ignoring the fact that there's probably a channel on this side. And actually this should be like quite close together. There's a gap here that doesn't really exist. Or there's interstitial fluid, right? There'd be some fluids here, but you would have to have protein. Like this we drew very small. In reality, if this is a cell, it should be the same size as that cell, or close to. So this would have its own proteins that would allow things.
It's gonna have like similar.
I don't want to say similar structure because it's a different type of cell. It's an endothelial cell instead of epithelial, but it'll have a protein that allows it through. Yeah, so you could say that like somewhere here there's a small green transport protein there. Okay, so step-by-step process. Create a fake gradient. We have to input energy to do that. You could even add here that this uses ATP, right? ATP. HL's ATP breaks down to what? ABP and P. Good. SLs, don't worry about that. But ATP is required. We create this low sodium gradient inside the cell, which drives sodium to move down the gradient from high to low, from lumen into the cell, which, using the co-transport protein, brings glucose with it. And once glucose is inside the cell, it doesn't like being in this high concentration area, so it will diffuse out through the glucose channel, eventually ending up in the blood capillary inside the villus. Now, before we wrap things up, let me just check. Oh, wouldn't it be nice if we could go to the lab and we could do a little experiment to model how the small intestine can be sort of replicated using objects in the lab. Hopefully someday. Okay. We have experiments that allows us to do this. The really nice thing about this is it can be a really simple idea for an II, right? If you're interested in things like nutrition, digestion and absorption, you could do a test where you model what happens when you're digesting different types of foods, right? And then how that might be impacted in different conditions. So it can be pretty easy, simple. You get a lot of trials. Still shows some personal engagement and so on. Okay, so yes, modeling in science, an important kind of first step in any experimentation, right? If you can't do it in the human or in the animal model or in the live model right away, live organism right away, using a model might be your next best thing, or at least your first step towards that. Okay. Last point of interest that I wanted to mention. Why do you think, no, let me ask this differently. Athletes are suggested, recommended, if they are dehydrated, to not drink water, but to rather drink sports drinks like Gatorade or Pocari Sweat or something like that. Why do you think that is? Because what?
Electrolytes.
That is correct. What is an electrolyte? You don't know. What are electrolytes?
Ion.
Ions. Why do you suppose that drinking something that has ions in it is more beneficial than drinking straight water?
Because it's increasing the gradient of sodium ions in the room.
That's one thing. Yes. Yes, true. You create a high sodium gradient if the ion we're talking about is sodium. Yeah. What else? If you're playing a sport, if you're working out, or whatever, what are you losing?
Water.
Aside from water?
Sodium.
Aside from sodium? Glucose. How do you get glucose back? With
Like teasing yourself.
with sodium, right? So yes, you need to consume some sugars, but just sugar won't be enough. It won't. Remember, we said glucose can't enter by itself. Water is just water, right? Water is not going to give you energy. It'll help replace the water you're losing. But if you run out of glucose for respiration, water's not going to help you, right? If you taste Gatorade, right, what does it taste like? It's a little bit salty, right? It's a little bit weird. It's not like juice or soda, like pop. It has like a bit of a salty kind of flavor to it. And if you want to go on like a deep dive, I highly recommend. There's a documentary, I guess, of like the people who discovered. It's like half discovery, half invention of Gatorade. I feel like I maybe mentioned this last year. I'm not sure. But they were, so it's, anyone know where Gatorade was invented? Or where, no? No? So it's the University of Florida. Anyone know anything about the University of Florida? What's the mascot of the University of the Gator. Yeah, the Gator. It's called Gator. Yeah, Gatorade. It was made for their football team. Okay, so there's a professor there who was doing like, he was actually a, like not related to sports at all. He was in like the medicine, and he was doing like kidney stuff. He was a kidney expert basically, and analyzing like urine samples and blah blah blah. And then the football team, like coaches were like, all of our athletes, like when it's really hot, they're just done. Like they like faint, or they can't go like for very long, and so they all came together to try and figure out like the different absorption. Basically, he'd like put athletes, they'd do their practice, and then he'd test their urine to see what was in the urine, and he'd like give them like different mixtures, like that he knew the concentrations of. They'd go to practice, and then he'd test their urine after to see what was there before and what got absorbed. And that's how he came up with Gatorade. That before this, they didn't know that there was this cold transport. Nobody thought to put salt, right? Why would you think salt would be required? Salt dehydrates, right? Salt takes water out. Right? It's counterintuitive. But here he realized that by adding salt and sugar at the same time, the sugar actually is helped to be absorbed. All right, thanks, people. See you this afternoon.
Michael's muted.